Welcome to the Explainer. Today, we're diving deep into the fascinating macro to micro battleground of rheumatoid arthritis. It's really a look at how this disease impacts the whole patient and exactly how modern medicine fights back, literally at the atomic level.
To kick things off, we really have to ask the fundamental question driving all of this incredible research. How do you actually stop the human body from attacking its own joints? How can we intercept the immune system when it launches friendly fire against itself? The answers? Well, they lie in this brilliant, complex combination of chemistry, biology, and holistic care.
Here's our roadmap for today. We'll start with understanding RA, move into foundation therapies and the anchor drug, then explore the biologic revolution, targeted synthetics, and finally, look at the holistic future.
All right, Section 1.
Understanding rheumatoid arthritis.
So it's absolutely crucial to understand that RA isn't just some simple joint issue. It's a chronic, progressive, and systemic disorder. It specifically targets the synovial joints, sure, but it's driven by these rogue immune cells and elevated nocturnal pro-inflammatory cytokines. Because these inflammatory signals surge at night and they aren't fully suppressed by the body's natural cortisol, well, patients wake up feeling the absolute brunt of this cellular warfare. And this systemic malfunction, it is incredibly widespread. We're talking about up to 1% of the entire global population being affected. And what's deeply important to note here is the demographic disparity. This disease is actually 3 times more prevalent in women than in men. This tactical assault on the body results in severe, prolonged morning stiffness, mostly due to those nocturnal cytokines we just mentioned. But because it's a systemic disease, the damage goes way, way beyond the joints. Patients experience extra-articular features, which basically means outside the joint, like profound fatigue, anemia, and even widespread systemic damage. We're talking vasculitis, pericarditis, and a significantly increased risk of osteoporosis.
Moving right along, Section 2, Foundations of Pharmacologic Therapy.
Now, take a look at the molecular blueprint of prednisone. It's a classic corticosteroid deployed to fight severe inflammation, and it's often used as a bridge until longer-acting drugs kick in. But the real magic? That happens when medicinal chemists actually start tweaking this exact structure. So here is the crucial point. By simply inserting a C1 to C2 double bond into this steroid skeleton, medicinal chemists drastically amplify the drug's anti-inflammatory superpowers. Not only does it increase glucocorticoid potency and metabolic stability, but it actually reduces unwanted mineralocorticoid activity at the same time. It's just a perfect example of how one tiny structural change yields a massive clinical advantage.
Okay, Section 3, Methotrexate, the anchor drug.
Methotrexate is, without a doubt, the undisputed anchor drug of the DMARD family. That stands for disease-modifying antirheumatic drugs, by the way. It doesn't just mask the pain, it actually interferes with the disease process itself, and it does so with an incredibly favorable balance of both efficacy and cost. Now, if we compare natural folic acid with our therapeutic weapon, methotrexate, they look almost identical, right? At first glance, anyway. But if you look a little closer, it's the really subtle structural differences that change the game entirely. Notice how folic acid contains an OH group and an NH group. Methotrexate actually replaces these with an NH2 and an N-CH3 group. These 2 tiny substitutions give methotrexate the power to tightly bind to and inhibit the DHFR enzyme. And by doing this, it essentially cuts off the metabolic supply lines required for DNA synthesis. It literally stops rogue, proliferating lymphocytes right in their tracks. However, we have to be incredibly careful with a tool this powerful. You see, methotrexate is excreted from the body unchanged via specific renal transporters, known as HO8T1, 3, and 4. But if a patient takes NSAIDs, common painkillers, those NSAIDs actually inhibit these exact transporters. The result? The body can't clear the methotrexate, which leads to a really dangerous toxic accumulation. It's a critical interaction that every single pharmacist and patient absolutely must watch out for.
Which brings us to Section 4, the biologic drug revolution.
Over the last few decades, there has been an absolute explosion of targeted therapies. Before the late 80s, options were pretty limited. But beginning with the TNF inhibitors like etanercept in the late 90s, we see this rapid evolution of targeted biologics and synthetic drugs entering the arsenal. It completely changed the landscape for patients who just don't respond to standard DMARDs.
Taking a look right inside the cellular environment of the synovium, we can see exactly where these biological defense systems intercept signals. The goal? To stop the ultimate enemy, osteoclasts, causing bone resorption. Notice how these different drugs act almost like highly specific roadblocks at various points in the immune cascade.
To break that down a bit further, each biologic has a highly specific mission. For instance, rituximab acts like a sniper, binding to CD20 to actively deplete problematic B cells. Meanwhile, abatacept competes to block the vital CD80 and 86 pathways, which is essentially the required second signal needed for T-cell activation. And tocilizumab? It directly blocks the IL-6 receptor. They aren't just broad suppressors, they are precision instruments disrupting the inflammatory network.
All right, on to Section 5, Targeted Synthetic Therapies.
Take a look at abrocitinib. It's a small molecule designed to selectively target and inhibit the JAK1 signaling pathway. Because it's a small molecule, it actually works inside the cell, blocking the kinase enzymes that transmit those cytokine signals we talked about earlier. But, as you might guess, engineering this kind of selectivity is incredibly difficult.
What's really fascinating here is the sheer precision required. During the drug's development, researchers actually found that modifying the structure with a 1,3-trans-cyclobutyl group reduced its potency. Think about that for a second. This reveals just how incredibly tight the spatial or steric requirements are for the JAK1 ATP binding pocket. A molecule literally must be engineered flawlessly to fit the lock and neutralize the target, all without accidentally hitting other pathways like JAK2.
And finally, Section 6, Holistic Care and the Future. Ultimately, controlling RA requires a cohesive squad working together. You need pharmacists rigorously counseling patients on DMARD compliance and monitoring for toxicities like we saw with methotrexate. You need physical therapists utilizing exercise and thermal therapies to maintain joint mobility. And, as a last resort when joint destruction gets severe, orthopedic surgeons stepping in. It is a true multidisciplinary effort from start to finish.
As we close out this explainer, we really have to look to the horizon, because despite all these incredible molecular tactics, a mortality gap still exists between RA patients and the general population. So the final question is, will future advancements in customized, personalized gene therapy finally end the friendly fire entirely and close that mortality gap for good? It's a thrilling frontier for sure, and one we will certainly keep our eyes on. Thanks for joining me on this deep dive.